Hey everybody, welcome back to the third lecture. So now we're going to be looking at the types of mutations, and we've already talked about these. I've already given you the definitions. In the next slide, we're looking at this codon table again. Remember that we read our DNA 3 nucleotides at a time, and so these are the RNA nucleotides, but they would be corresponding to the DNA. So wherever we have in our DNA a T, we would have a uracil instead. Where we have a C, we would have a C in our RNA. If we have an A, we'd have an A in our RNA. And if we have a G, we'd have a G in our RNA. And I also want you to see that the codon table codes for 20 different amino acids, but there are 61 different codons. And so there's a lot of different codons that code for the exact same amino acid. So like here's proline, CCU, CCC, CCA, and CCG all code for proline. Here we have 2 codes for histidine. Here we have 4 codes for arginine and 2 more codes for arginine. So there's a lot of different codons that code for the same amino acid, which means that not all mutations are going to cause changes in the amino acid or in the protein production or amino acid sequences. So here we have the different mutations that we were talking about. This first one shows a silent mutation, which means there's no effect. So you're producing the same amino acid. And so this is what the wild type DNA looks like. This is what the wild type RNA should look like. And this is what we're producing. Okay, this is the translation. Over here, we have a mutation in the DNA. So where over here we have an A, we have a T put in. And so instead of a GUA, we have a GUU. But GUU and GUA both code for valine, so we don't change the amino acid sequence, which means the protein doesn't change at all. That's a silent mutation. A missense mutation is going to result in a change in the amino acid, and that's going to change the functionality of the protein. So structure determines function. If the structure changes, the function can also change. And so here we have a change in our third amino acid, the first letter of that third codon. This is the wild type codon. This is the wild type messenger RNA, and this is what is supposed to be produced. But instead, over here we have, instead of the C, we have a mutation where there's an A there. And so now we have an A in the amino acid sequencing. And so CCC codes for proline, but ACC codes for threonine. So now we have changed the amino acid sequence, and that can change the functionality of the protein. Oh, sorry. Whoa, okay. Stop. Don't touch anything. All right. So now let's look at the nonsense mutation. So a nonsense mutation ends up coding for a stop code, which means we stop protein synthesis. And so here again we have that wild type. Here's the messenger RNA wild type, and here's what we're supposed to produce. But over here, at this last spot, we, instead of having the C, which TAC transcribed as UAC, produces tyrosine. Now we have TAG transcribed as UAG, and that codes for a stop protein. And so when you stop protein synthesis, the protein becomes non-functional. So you don't have the amino acid sequence you're supposed to have. The last type of mutation that we're talking about is a frameshift mutation. And so this is caused by either having an insertion or a deletion of one or more nucleotides. So here is the wild type DNA again. Here is the wild type messenger RNA, and here is the series of nucleotides that are supposed to be produced. But in this case, we are removing the T and A, so this would be a deletion mutation. Now you have A, G, C, G, but then we skip these two and we just have CCC. So what happens is you're going to change the reading frame. That's why it's called a frame shift. We change the reading frame. So we normally read 3 nucleotides at a time. These three go together, these 3 go together, these 3. So we still have that first 3 nucleotides together, but the second 3 nucleotides have changed. So now instead of reading GUA here, now we're changing and we're taking 2 of these, we're shifting it over, and we're reading GCC. So we've changed the complete sequence of amino acids after that frame shift. So everything down from that frame shift is going to be changed. And so let's look at a frame shift that you can understand probably a little better. So I know you can read this sentence, and I would read it out loud because that's what I usually tell my students to do. The cat ate the rat. So we are pretending that these are nucleotides, and we are reading our nucleotide codons 3 letters at a time like we're supposed to. So the cat ate the rat, that is a very obvious, complete sentence. It makes sense. We understand what's going on. If this was a protein code, we could produce that protein, right? But now let's take and delete an A. All right, so we're going to have everything else still here. But now read the sentence. The cat tet her at. And that at isn't even complete because you no longer have the enough nucleotides. So this doesn't even code for a codon. The cat tet her doesn't make any sense though, right? So that is a deletion mutation that causes a frame shift. Here's another one. So now in this one, let's add an A, okay? This will be an insertion mutation. So now we're adding an A. So now it says the cat at eth era ch. And again, does not make sense. So everything down from that mutation, that frame shift, is going to be different and likely will code for a stop codon very early. And so you will have a non-functional protein. Mutations can occur spontaneously while DNA is replicating, but this is not very common. So DNA replication is a very regulated process, and there are a lot of different mechanisms that we use to protect our DNA. More often, we have induced mutations. So induced mutations occur when a mutagen of some sort stimulates the change. A lot of times mutagens can also be considered carcinogens. Not always, but carcinogens are cancer-causing materials. And almost all carcinogens are also known as mutagens. So there are a lot of different mutagens out there. There are chemical mutagens. There's also UV light or radiation mutagens that are out there. So not just UV light, X radiation, gamma radiation, those all cause mutations, and they're considered mutagens. Okay. So what are some of these types of radiation that we have? Ionizing radiation, so this would be like gamma rays or X-rays. These are going to break the backbone of DNA and cause the DNA to basically break up into little pieces. Whereas non-ionizing radiation or ultraviolet radiation causes thymine dimers. So a thymine dimer occurs when 2 thymines are on the same strand of DNA next to each other, and instead of binding with their complement on the opposite strand, they bind together. And when that happens, then when you are replicating, you can't replicate properly. And so this can cause frameshift mutations or sometimes point mutations. But we have a lot of mechanisms to repair these different mutations that can occur. So DNA polymerase has proofreading ability, which means it will read through, it produces the new DNA strand, the daughter strand, but then it also proofreads to make sure that everything is proper. There are mismatch repair enzymes that are going to kick out the damaged DNA, the DNA that has any damage to it, and then allows DNA polymerase to come in and build a new set of DNA there. So this is your ionizing radiation, so like X radiation or gamma radiation, causing breaks here, here, and that can break the entire DNA strand up. And then this is what a thymine dimer looks like. So you have that thymine dimer between two nucleotides, two thymines on the same strand of the DNA. So DNA polymerase proofreads the DNA, and when they do this, they actually will kick out improper nucleotides. So if the wrong nucleotide comes in, DNA polymerase will just kick it out and the proper nucleotide will come in. Mismatch repair, mismatch repair enzymes will quickly fix those small mistakes. But we also have dark repair enzymes and light repair enzymes. So dark repair enzymes are enzymes that work under dark environments. So they are activated in the dark. They don't work during when there's a lot of light in the place. So like if you put bacteria in a window to grow with that sunlight shining on them, dark repair enzymes won't work. But if you put them in a dark environment where no light can get at them, then those dark repair enzymes can work. That's what a dark repair enzyme is, okay? And it recognizes those changes in the DNA, and it'll cut out the incorrect pairing or the damaged region, and then allows DNA polymerase to produce a new template strand that can be used. So this is what we use when we have thymine dimers formed with UV radiation. And then we have light repair enzymes or direct repair enzymes, which use photolase enzymes, which are going to be usable during light environments, okay, with the visible light. And these will break apart those thymine dimers so that the DNA can then be replicated properly. And so here we have a thymine dimer from UV radiation. So what will happen is you're going to have the original strand here, and then you have this complement up here. These two aren't bound. So we have these light enzymes that come along, and they will break apart that thymine dimer so that the thymine dimer now is able to bind properly. And DNA polymerase will recognize this mutation and cut out the damaged region, and then DNA ligase helicase will cut out the material. DNA polymerase will then add new nucleotides, and then ligase will seal everything together. I'm going to stop here on this video. We're almost done, but we do have a little bit more, and this next process is going to probably take me about 7 or 8 minutes, and I don't want this video to get too long. So I'll talk to you guys in a little bit. Bye.